Welcome to the lesson on mutations. In this lesson, we're going to be discussing single-base mutations, such as point mutations and frameshift mutations, and we'll also be discussing chromosomal mutations, such as deletion, translocation, and inversion. All right, let's go ahead and get started. We're going to start with single-base mutations, which means changing one base in the sequence. We're going to talk about two kinds of single-base mutations, and the first kind we're going to discuss is point mutations. A point mutation is when one base is changed to another base. So, for example, suppose that mRNA codon CCG became CCA, so that G became replaced by an A. Well, if you take a look at your circular table, this would end up coding for the same amino acid, so there would be no effect. In this case, the amino acid would be proline if you were to look it up on your circular table. So CCG produces proline, but so does CCA. Since they both produce the same amino acid, this is called a silent mutation because there is no net effect on the changing of the protein here that's made. However, suppose the mRNA codon GUA became UUA. This would code for a different protein, different amino acid, therefore changing the protein that is produced. GUA would produce the amino acid valine, while UUA would produce the amino acid leucine. And so you can see that changing the G to a U would have a pronounced effect, changing the amino acid, therefore changing the protein. Let's look at a pretend example here. And the pretend example is, the boy had the big dog. Now, of course, it's pretend because there's a lot of letters in here that aren't letters in the mRNA codon sequence. But nonetheless, for the boy had the big dog, suppose the E got changed to a Q. This would be an example of a point mutation. A real example of this would be sickle cell anemia, which we've talked about in class already. This is where some of your red blood cells take on a different shape, and it affects their ability to hold oxygen because of their surface areas being reduced. Okay, another type of single base mutation is called a frameshift mutation. And this is one in which one base is dropped or lost. Look at our example again. The boy had the big dog. Now suppose the E got dropped. The E from the got dropped. So everything shifts to the left one. So our first codon here would be THB. And then everything shifts once beyond that. So OYH, then ADT, etc., on down the line. A real example of a frameshift mutation is Tay-Sachs disease. And this results in big changes and problems, as we've talked about with this disease already in class. All right, now let's discuss chromosomal mutations. Chromosomal mutations, as their name implies, is damage to an entire chromosome in some shape or manner. The first kind of chromosomal mutation we're going to discuss is deletion. Deletion is the loss of a whole chunk of a chromosome. This is often extremely deadly and damaging. A real example of this is Cri-du-Chat syndrome, which is a syndrome that takes place in the respiratory system. Okay? Next type of chromosomal mutation we'll take a look at is translocation. Translocation is moving a piece of a chromosome onto another chromosome. This may or may not be damaging. It depends on the importance of the chromosome. A real example of this is Burkitt's lymphoma. And you can see the side of the face very distorted in this picture right here. The third type of chromosomal mutation we're going to discuss is inversion. In inversion, a chromosome breaks and the broken piece reattaches backwards. And this, as you can imagine, causes major problems. All right, let's take a look at another video in which our amoeba sisters are going to cover some of this, in which many of these mutations are discussed and produced in animation.